Hi, I'm Stefan Bertani from the laboratory of Dr. Frank Zauer in the Department of Biochemistry at the University of California, Riverside. Today, we will show you a procedure of chromatin immunoprecipitation. So, let's get started.
This procedure begins with formaldehyde cross-linking of human embryonic stem cells. Prior to cross-linking, human embryonic stem cells are thawed from liquid nitrogen for 5 minutes at 37 degrees Celsius and then washed with media. The cells are then plated on gelatin-coated 6-well plates and allowed to expand for 2 to 3 days. For each immunoprecipitation reaction, we will need to start with 50 to 100 million cells. The cells are first trypsinized and collected in suspension. Fresh formaldehyde is added to each cell suspension at a concentration of 1% and incubated for 10 minutes at room temperature. Next, add 1/10 volume of 1.25 molar glycine to quench the formaldehyde and incubate for 10 minutes. Rinse the cells twice with 10 milliliters of PBS and then pool them in 50 milliliter conical tubes. Spin the tubes in a centrifuge at 1000 RPM for 5 minutes at 4 degrees Celsius in order to collect the cells in a pellet. After the spin, discard the supernatant and resuspend the pellet in 1.5 milliliters of lysed buffer. Finally, transfer the cells into a 1.5 milliliter microfuge tube. After the cells have been transferred, flash freeze them 3 times in liquid nitrogen and then use a tissue grinder to break them. Once the cells are cross-linked, they are ready for immunoprecipitation, or they may be stored frozen at -80 degrees Celsius indefinitely. Once you have cross-linked the cells, you need to prepare the magnetic beads. To do this, add 50 microliters of dinyl beads to 1.5 milliliter low retention microtubes. Set up 1 tube of beads per immunoprecipitation. Then add 1 milliliter of block solution. Now, collect the dinyl beads using dinyl MPC. Place the tubes in the magnetic rack. Allow the beads to collect on the side of the tube. This should take about 15 seconds. You can invert the rack twice to help collect beads. Once the beads have been collected, you can remove the supernatant with pipetter. After the supernatant has been removed, add 1 milliliter of block solution and gently resuspend the beads by removing the magnetic strip from the rack and inverting the rack with the tube still in place. You can do this 10 to 20 times or until the beads are evenly resuspended. Collect the beads as you did previously. Once collected, you can remove the supernatant with a pipetter. Repeat this wash using 1.5 milliliters of block solution. After the second wash, you can resuspend the beads in 750 microliters of block solution, and then add 10 micrograms of antibody. In our research, we use an antibody against the protein MLL. Incubate the mixture at 4 degrees Celsius for a minimum of 6 hours or overnight on a nutator. During this incubation, you may sonicate your cross-linked chromatin, which we will demonstrate shortly. After the overnight incubation, remove the antibody solution, and then wash the beads 3 times using 1 milliliter of lysed buffer, as you did previously, using the magnetic rack to collect the beads. After the washes, you are ready to add your sonicated chromatin. Before beginning sonication, you first need to retrieve the frozen cell pellets from the -80 degrees Celsius and allow them to thaw. Prepare a 15 milliliter conical tube for sonication. This is done by cutting the tube in 2 pieces at the 5 milliliter mark and discarding the upper half. The tubes can be covered with parafilm or the tube cap while setting up. Using a tube rack, position the tube so that the sonicator probe sits approximately 0.5 to 1 centimeter above the bottom of the tube. Be sure that the probe is centered and does not contact the side of the tube to prevent foaming of the solution. Now, sonicate the suspension for 6 rounds of 20 seconds each. Samples should be kept in an ice water bath between rounds of sonication. To decrease foaming, initially set the power output to 0 and then manually increase to the final power during the first burst. After sonication, transfer the material to a 1.5 milliliter tube and then spin at 13,000 RPM for 10 minutes at 4 degrees Celsius to pellet the debris and harvest the supernatant. Transfer the supernatant to a new 1.5 milliliter tube. Save 50 microliters of the cell lysate from each sample as input DNA. This will be used later to check the size of the sheared chromatin. This can be stored at -20 degrees Celsius. At least one input DNA aliquot should be kept per batch of sonicate lysate. Now we are ready to immunoprecipitate the chromatin. To immunoprecipitate the chromatin, first add the chromatin equivalent of 50 to 100 million cells from the supernatant collected in the previous step to 50 microliter antibody magnetic bead mix that we prepared earlier to a final volume of 1 milliliter. If necessary, this volume can be adjusted with lysed buffer. Invert to resuspend the beads. Incubate this mixture overnight on the nutator at 4 degrees Celsius. After incubation, collect the beads using dinyl MPC. Place the tubes in the rack, allowing the beads to collect on the side of the tube. This should take approximately 20 seconds. Afterwards, invert the rack twice to help collect all the beads. Then, remove the supernatant with a pipetter, changing tips between samples. Next, we need to wash the beads. To do this, add 1 milliliter of lysed buffer to each tube and gently resuspend the beads. This can be done by removing the magnetic strip from the rack and inverting the rack with the tube still in place, about 10 to 20 times or until the beads are evenly resuspended. Afterwards, collect the beads by replacing the magnetic strip. Remove the supernatant by pipetter. Repeat this wash 3 to 5 more times. Remember to change tips between washes. Next, wash the beads 4 to 6 times in 1 milliliter of IP1 buffer, as just described. Then, similarly, wash the beads 4 to 6 times in 1 milliliter of IP2 buffer. Finally, wash the beads 4 to 6 times in 1 milliliter of TE buffer, pH 8.0, as done with the other washes. Remove the TE after the final wash. Now, spin at 3000 RPM for 3 minutes at 4 degrees Celsius to remove any residual TE buffer. The beads are now ready for elution. To elute the chromatin, first add 210 microliters of elution buffer and elute the material from the beads by incubating the tubes in a 65 degrees Celsius thermal mixer at 800 RPM. Incubate for 15 minutes or up to 30 minutes to improve recovery of the eluate. Spin down the beads at 13,000 RPM for 1 minute at room temperature. After centrifugation, remove 200 microliters of the supernatant and transfer to a new tube. And then add 200 microliters of TE, pH 8.0. Now our samples are ready to decross-link. To reverse cross-link the immunoprecipitated DNA, incubate the supernatant in a 65 degrees Celsius water bath for a minimum of 6 hours and a maximum of 15 hours. This incubation can be done in a water bath to minimize condensation. Next, thaw the 50 microliters of input DNA that was reserved after sonication. Reverse cross-link this input DNA by incubating it at 65 degrees Celsius, as was done with the eluate. Now we are ready to purify the DNA. The DNA is then purified and precipitated using phenylchloroform extraction, followed by ethanol precipitation techniques. The precipitated DNA pellets are resuspended in water and then can be stored at -20 degrees Celsius for future use. The purified input DNA can be run on an agarose gel to check the size of the sheared chromatin. Ideally, the chromatin should be between 250 and 750 bases.
So, we have just shown you how to do a chromatin immunoprecipitation from human embryonic stem cell. When you're doing this experiment, it's important to use a high affinity antibody, chromatin with the right size range, and a high stringency wash steps. So that's it. Thanks for watching and good luck for your experiments.